All right, thanks for having me. It's been great. It's my first GET event, and I was here yesterday and today, and it's been very interesting and fun for me. I'm going to tell you about a new OME that's coming along. You know about genome. A genome is the complete set of DNA and all the genes. An immunome is a term that we are using for the complete set of recombined immune receptor genes. So you'd think that if immune receptor genes are a subset of the genome, that collection would be much smaller. But in fact, that's not the truth, because these genes recombine. And so the human genome is something like 10 to the 4th genes. But the human immunome, we don't know how large it is. And that's the project we're going to embark on, is to define how big and to explore it. But it could be many, many orders of magnitude larger than the genome. So this is the largest sort of sequencing escapade probably that anyone has embarked on. So to remind you how antibodies and TCRs, for that matter, are encoded, there is a set of variable genes and diversity genes and joining genes and even constant genes. So you select one of your 51 heavy chain variable gene segments, one of your Ds and one of your Js. The first event is to recombine using RAG genes D and J together, and then you add a V. So now you have a single V, D, and J selected from this collection in your genome, and they recombine. So right there you get about 10 to the 4th combinations. This is called combinatorial diversity, and that's about the size of the genome right there, just the combinations. But it's also interesting, when you join these, they're not perfect joins. So there is a set of enzymes and isoforms that either nick off a nucleotide or add on, and they can do several or many. And you'll remember from the central dogma of DNA to RNA to protein, if you add one nucleotide, you change the reading frame. And so if you put a single nucleotide in between your D or your J, your V and D and J, you completely change the reading frame. So you get an enormous boost in your diversity at that point, just because of the joining, and that's called junctional diversity. At that point, you're at 10 to the 11th potential combinations just by putting things together. And during an immune response to an infection or vaccination, the whole point of that process is to induce somatic hypermutation. So in most genes, you avoid mutating them. You don't want mutations in your genes. But in the antibody locus, this is highly allowed. So the process of somatic hypermutation allows DNA strand breaks by a set of enzymes, and the polymerases that repair that are error-prone. So you allow mutations to occur and accumulate. So every position of the variable gene can be mutated to any other nucleotide. There are some biases and motifs, but basically you can put any set of mutations in there. So this is the recombined gene, and we have no idea how many of these a person has, and so on. And these are the genes, but if you look at the protein that's encoded, there's a heavy chain and a light chain, and so it's sort of like you have two hands that come together. And the variability that you get in these genes is out in the tips, and this is what encounters the microorganisms such as viruses or bacteria. And one of these genes is encoded by the junction, where 3 genes come together and all those non-templated nucleotides. So this loop right here is very variable structurally and in sequence, and that's basically how you recognize an infinite number of threats to yourself. In fact, we can make chemicals that have never existed in nature and inject you with it, and you'll make an antibody to it. So the structural diversity of this little loop, the heavy chain CDR3, is enormous, and that's the key to vaccination and immunity by antibodies. If we take all of your blood that we can out of your body and sequence all of the antibody genes that are being expressed at the time, it's difficult to look at. We're lacking proper visualization tools. This is one way we're doing it right now, called a Circus Plot. So the variable genes I mentioned, here's your 51 or so in color along the right side, and then there's an inner circle with the 27 Ds, and then there's 6 Js I mentioned. So we're connecting V, D, and J, and each combination of the 10,000, that's more than 1% in this person's genome or immunome, is shown in color. There's some gray ones underneath for the other 10,000. And you can see they're different sizes. So you have different representation of each of these combinations. And this is just a single sample. In fact, it's just a subset of the blood, because in this person, we sorted out cells that had B cells that had never seen an antigen and B cells that were memory cells that had previously seen an antigen, and there's an intermediate type that has not class switched from IgM to IgG. And if you look at these patterns, this is just from a single person, you can see the subsets have different patterns, and there's actually a different pattern. You can see there's lots more little gray ones underneath there that represent the diversity. So the diversity changes within a person after antigen exposure. And we think, what would it be like if we were able to get to the very bottom of people and say, how many of these do people actually have in them? How do they change over time? What if we immunized you? How would it change? What does it look like when you're born in your cord blood and then a year later and then when you're 100 years old? What does it look like when you get up in the morning, every minute, when you're sleeping, when you run an ultramarathon? There's just so many influences you can imagine when you eat. Much of the discussion you just heard probably influences these immunomes. And we're just getting these first snapshots of what a normal immunome would look like. So the Human Immunome Project, the goal is to sequence all of the antibody and T-cell receptors on the planet. That's the idea. The number could be as much as 10 to the 15 or some absurd number like that. We don't think it'll be that large because we think things will be missing. So short term goal is how many sequences does a person possess? Are they stable over time? If you sequenced my immunome and your immunome, would we share sequences or would it be completely different? Could you recognize me just based on my antibody sequences? When we're immunizing, we think we are interrogating the common or the shared repertoire. And then, of course, age and all these other variables. And what's missing will also be interesting because during negative selection, during immunontogeny, you want to get rid of things that bind to your brain and other essential organs. So we believe, our hypothesis is, most of the immunome that could be there will not be there because of these negative selections, and disease processes may bump you into those forbidden areas. So what's missing might be interesting. And so that's sort of the equivalent of the Human Genome Project. There are also applications you can imagine, perturbations like a vaccination. We also think the ultimate future of this is to understand a complete catalog of all the structures and what those antibodies do. I'll show you an example. This is a person that we followed for several years, and we've been sequencing the person's immunome, not at complete death, but, and this is an antibody clone to HIV at 9 different time points. And you can see this is a phylogeny. We're getting mutations in this clone. One side clone wanders off and explodes, but over 9 time periods, the clone progresses and mutates. And what's amazing now is synthetic biology. We can make all these antibodies in the lab. It's like having a 3D printer for antibody proteins, and we can go and say, do these kill HIV? So we've actually started to do this. So that's an application where we think you can make point-of-care antibodies based on a catalog on a computer. And we also think we can look at these sequences, predict the structure, and understand their function. So there are technologies like this. Rosetta is a three-dimensional software. So what we're doing here is running sequences over the structure of a known antibody that kills HIV, and each one we can sort of energetically minimize and predict. So we've just published a paper this month where we looked at 500 million sequences from people who we know did not have HIV. We got their blood from the Red Cross, and they'd been fully tested, and we looked around and said, can we find any antibodies in there that bind and kill HIV? And we did. So we can recognize predicted structure and function just having millions of sequences. So that's the future of this. And the idea is to do 1 donor, 2 donor, 1,000 donors, and eventually we think we should be able to collect the global repertoire. Lots of folks do this. These are the folks who did the data I showed you directly. This is my group, very diverse group at Nashville, and the folks that give us money. So thanks for listening. I think Jason said we could ask a few questions if you want.
Are you doing B and T cells or just T cells?
Yeah, I emphasized antibodies because they also mutate and have indels, and that's also my field of expertise. We also are doing T cells. T cells are different in that they don't mutate, but they probably out of the gate have a broader repertoire. So we will be doing T-cell receptors as well.
And are you looking at the antigen reactivity besides modeling, or just?
Well, so in my day job, we make virus-specific or toxin-specific antibodies and sequence those, and we sequence the repertoire, the same sample, and build those phylogenies that I was showing you. And we think that we can attribute specificity increasingly by doing that type of one-by-one work, looking at somatic variants of a known antibody if we ascribe that specificity in the immunome. But it'll be a while before we can ascribe specificity to even a significant portion. But that is a goal, yeah.
Can you say anything about autoimmune disease and what your research will contribute to that?
Yeah, this is a very interesting area, autoimmune disease. So you would think that you should not have antibodies that would bind to yourself and harm yourself, and the immune system is made to select against those. But when we look, people do have those. And in fact, some of these antibodies are encoded in the germline genes. We have found antibodies that bind to sugars on human cells and so on. And it may be that ultimately understanding which of these antibody sequences bind to your own proteins, we can make therapeutics that block that. We could even, we're doing projects now to make antibodies against antibodies. Jern got the Nobel Prize for that. Anti-IDs, we think we might be able to make anti-IDs that block the autoimmune ones if we understand which ones they are. And again, we can make them and then make antibodies to them. All right, thank you.